Ribosomes and protein synthesis. How do you read the language of DNA? Would you read the bases one at a time like RNA? Or would you set a code and treat them as words? The genetic code is read 3 letters at a time so that each word is 3 bases long and corresponds to a single amino acid. The first step in decoding genetic messages is to transcribe a nucleotide base sequence from DNA to RNA. This transcribed information contains a code for making proteins. Proteins are made by joining amino acids together in long chains called polypeptides. As many as 20 different amino acids are commonly found in polypeptides. The general structure of an amino acid is the amino group and the carboxyl group, and a unique R group joined together. For instance, the amino acid and the carboxyl group here is joined by CH3. This is alanine joined together with serine with a peptide bond. The specific amino acids in a polypeptide and the order in which they joined determine the properties of the different proteins. The sequence of amino acids influence the shape of the protein, which in turn determines its function. RNA contains 4 different bases: adenine, cytosine, guanine, and uracil. These 4 bases form a language or genetic code with just 4 letters, A, C, G, and U. Of course, the U is a replacement for thymine in the DNA. Each 3-letter word in mRNA is known as a codon. A codon consists of 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain. This is a codon of UAG, a codon of AAC, and a codon of UCU. Because there are 4 different bases in RNA, there are 64 possible 3-base codons. 4 times 4 times 4 is 64 possibilities in the genetic code. This circular table shows the amino acid to which each of the 64 codons corresponds. To read a codon, start in the middle of the circle and move outward. So the codon UAC is tyrosine. Most amino acids can be specified by more than one codon. For example, 6 different codons UUA, UUG, CUU, CUC, CUA, and CUG specify leucine. But only one codon, UGG, specifies the amino acid tryptophan. Tryptophan is here. It's U, G, and G, tryptophan. The genetic code has punctuation marks. The methionine codon AUG serves as the initiation or start codon for protein synthesis. Following the start codon, mRNA is read 3 bases at a time until it reaches one of the 3 stop codons, which end translation. Here's a stop codon. Here's a stop. So UAA is a stop. UAG is also a stop. And UGA is also a stop codon. What role does ribosome play in assembling proteins? Ribosomes use the sequence of codons in mRNA to assemble amino acids in polypeptide chains. The sequence of nucleotide bases in an mRNA molecule is a set of instructions that gives the order in which the amino acids should be joined to produce a polypeptide. The forming of a protein requires the folding of one or more polypeptide chains. The decoding of an mRNA message into a protein is a process known as translation. Messenger RNA is transcribed in the nucleus and then enters the cytoplasm for translation. Translation begins when a ribosome attaches to a mRNA molecule in the cytoplasm. This is the ribosome, and it's 2 portions. One attaches to the mRNA, And this other portion of the ribosome attaches to the tRNA. As the ribosome reaches each codon or mRNA, it directs tRNA to bring the specified amino acid into the ribosome. One at a time, the ribosome then attaches each amino acid to the growing chain. This chain is what's going to grow. Each tRNA molecule carries just one kind of amino acid. In addition, each tRNA molecule has three unpaired bases, collectively called anticodon, which is complementary to the one mRNA codon. UAG has a anticodon complementary in the tRNA, UAC. That is a code for methionine. The tRNA molecule for methionine has the anticodon UAC, which pairs with the methionine codon AUG. The ribosome has a second binding site for tRNA molecule for the next codon. If that next codon is UC, a tRNA molecule with a AAG anticodon brings the amino acid phenylalanine into the ribosome. UUC, the anticodon is AAG, and that brings phenylalanine as the next amino acid. The ribosome helps form a peptide bond between the first and the second amino acids. Between these two, methionine and phenylalanine. At the same time, the bond holding the first tRNA molecule is broken and releases that tRNA because its work is finished. That tRNA then moves into a third binding site from which it exits the ribosome. So this last one is the exit. The ribosome then moves to the third codon, this third codon here, where tRNA brings the amino acid specified by the third codon. tRNA would bring that matching anticodon to the codon of the mRNA. The polypeptide chain continues to grow until the ribosome reaches a stop codon on the mRNA molecule. So this ribosome structure, blue and tan, would continue down the chain of mRNA, adding in amino acids to this polypeptide until it comes to this stop codon, UGA in this case. When the ribosome reaches a stop codon, it releases both the newly formed polypeptide and the mRNA molecule, completing the process of translation. Ribosomes are composed of roughly 80 proteins and 3 or 4 different RNA molecules. These rRNA molecules help hold ribosomal proteins in place and help locate the beginning of the mRNA message. They may even carry out the chemical reaction that joins amino acids together. What is the central dogma of molecular biology? The central dogma of molecular biology is that information is transferred from DNA to RNA to protein. There are many exceptions to this dogma, but it serves as a useful generalization that helps explain how genes work. So in the nucleus, transcription is done to create an mRNA, and that mRNA comes out into the cytoplasm and with the help of a ribosome is translated into amino acid combinations, forming a polypeptide. Most genes contain instructions for assembling proteins. Many proteins are enzymes, which catalyze and regulate chemical reactions. A gene that codes for an enzyme to produce pigment can control the color of a flower. Another gene produces proteins that regulate patterns of tissue growth in a leaf. Yet another may trigger the female or male pattern of development in an embryo. Proteins are microscopic tools, each specifically designed to build or operate a component of a living cell. Molecular biology seeks to explain living organisms by studying them at the molecular level, using molecules like DNA and RNA. Gene expression is the way in which DNA, RNA, and proteins are involved in putting genetic information into action in living cells. DNA carries information for specifying the traits of an organism. The cell uses the sequence of bases in DNA as a template for making mRNA. The codons of mRNA specify the sequence of amino acids in a protein. Unique proteins play a key role in producing an organism's traits. A large variety of proteins are used across organisms. One of the most interesting discoveries of molecular biology is the near universal nature of the genetic code. Although some organisms show slight variations in the amino acids assigned to particular codons, the code is always read three bases at a time and in the same direction. Despite their enormous diversity in form and function, living organisms display remarkable similarity at life's most basic level, the molecular biology of the gene. What other life structure would work